and I'll be demonstrating Opus in a series of short informational videos where we'll look at various aspects of the program, what it can do, and how you utilize it in order to be able to get the best insight from an admittedly complex field, genomics, mingled with nutrition and into the form of a type of precision personalized medicine. Well, since this is the introductory film, we might as well start at the beginning, which almost always winds up being at this particular page, which is the splash page for Opus at datapunk.net/opus23. The thing you're going to want to do is log in, and this is basically going to be the result of putting in your email and a password that you have either been assigned or changed in your account settings, as you'll see as we go a little further into this. So the first thing we'd want to do is log in. Now, I am going to put in some information here that hopefully you will not see, and it will bring me to this screen. Now, Opus will ask you, well, in addition to welcoming you, it'll ask you if you want to upload a client. And you'll see here I've got 30 clients in my account. You normally can do it from this particular link here, or you can go up to in the manage part of the upper menu bar, and you can actually go to profiles and list and load profiles. I am going to pause the movie at this point and load a profile because you would be looking at an entire screen of patients of mine and things like that, and I would probably get hip or all upset. So one of the things we'll do here is as you can see here, I've uploaded a client of mine who's now available. The name Leolandro Mysterioso should satisfy even the most ardent patient privacy people. And we have that loaded now. So Opus now is available for utilization. All the various apps, all the various functions in Opus will be applied to this particular client. You'll see here at the upper bar, you're always told who's your active client because it's listed here in the breadcrumb trail. So there are some things that you can do with regard to Opus that sometimes helps it out. So for instance, there are certain things in Opus that cannot be computed effectively or reliably. And sometimes it helps also to just add additional data on the client. And so here's where you can put in other particular details that may have some particular aspects with regard to the client that it will give Opus a little bit more insight. You can put in pathologies, any number of different things. These should autocomplete. So here, if let's say we were going to give the person ulcerative colitis as a diagnosis here, ABO type. Sometimes ABO type can be computed effectively in Opus via SNPs, but there are certain types that regarding certain platforms that you can bring into Opus that may or may not have the required SNPs in order to be able to compute ABO type. So if you have access to that information through serological data or elements like that, you can put that in as well. And then basically, like everything at this point, since it is a form, you would then go ahead and save the edited profile information. So that is essentially the basic function of how to get your client into Opus. You would normally load a client into Opus by first purchasing a license for that client. And let's see if we can look at this here. Well, since I have an editor account, I don't have the option to purchase a client from my account. But basically, once you get a license, you have a slot that's free that you can assign to the client. And then you would have the opportunity to go to this screen, which would have you upload a new client profile. And you put in some basic information, and at that particular point then you would create the new client, and having chosen a raw data file from any number of the services that report in a way that Opus can basically utilize. And you can see those services here. Opus will take input from Diagnostic Solutions, Genomic Insights, Ancestry, the two most recent versions of 23andMe and Genos. Uh, it will require certain things with regard to your client's name, date of birth, gender, things like that. And this helps with regard to some of its calculations. If you feel like you want to anonymize the patient, Opus doesn't really require that the name be the name. I mean, you could give it your own code or whatever. However, Opus is behind a very secure firewall, and the encryption that's actually generated with regard to client data is very good. It's much more uh, extensive than even required by the HIPAA bylaws, which actually have minimal encryption requirements. But Opus uses very, very sophisticated encryption on its data. So you can feel good about using the actual name, but you don't have to. So I have loaded the client, as you can see here. So we want to basically at this point go ahead and work with the client. But I just wanted to say something else about the flexibility of Opus with regard to this. Opus allows you to mix and match various platforms. So let's say I bring in my client says, well, I had 23andMe done in 2008, and it turns out that they have V4, which is really one of the better versions of 23andMe in terms of the library of SNPs that they report. Well, you can load that in, and they say, well, I mean, I have other ancestry information as well. You can actually aggregate several platforms in Opus, and that just gives you better and better and better coverage. And I think that that's important because certain things, for instance, Ancestry has certain SNPs that they report that 23andMe doesn't. 23andMe has certain SNPs that Ancestry doesn't report. The most extensive, actually, is the Diagnostic Solutions Genomics Insight. That has just really better coverage with regard to the more clinically significant SNPs. So I think that's generally the direction I like to go in because that information is one single upload. However, with Opus, you can use an app called, I think it's called Blender. And yes, and what you can do here is append data. So in this case, you can see here from my client, the only service that we have loaded up until now is 23andMe V4. However, I have other available services that I could load. So for instance, and every time you upload a file into Opus, you must remember that this has to be a zipped file of the raw data text file that's given to you when you download the raw data from any of these services. So it not only has to be the text file, which could be, I don't know, 35, 40 megabytes. It has to be a zip version of that, so that when you're uploading the file, you're uploading maybe about a 5 megabyte file. So that serves to maintain a certain level of bandwidth. So sometimes we run into trouble because people try to upload PDF files of the data. That won't work. Or they don't have the zip version of the text file. But typically when you download the raw data from the service, it comes as a zip file. Although sometimes I believe the most recent versions from 23andMe sometimes do and sometimes don't. But that's easy enough to do. Just go into your file program in the Mac. It's I think it's called Finder, and find where the file you've downloaded the file to, and then right click on it and just select compress. And then basically, even if the file hasn't been given to you as a zip file, you can make your own version of that. And then basically you can upload the file. You don't have to tell Opus what company or reporting service. The Opus will actually analyze the file and get back to you and say, it looks like you've uploaded an Ancestry file, or it looks like it's a 23andMe. And you can see here it tells you a little bit about the current coverage. So what we're looking at here with these pie charts is the total amount of the high quality curated zips and curated SNPs in Opus, and the percentage of them that are currently being covered by the loaded services. So we can see here we have almost 4,000 human curated SNPs in Opus, and the 23andMe V4 version that was loaded for this client is providing information on 31 128 of those SNPs. So the coverage is about 80%. Would be nice if it was 100. Now, if I had ancestry data on this person, I could upload that, merge it with the current file that was there in 23andMe, and the total amount of SNP coverage would increase, the percentage would increase and get higher. Keep a couple of things in mind that the 3,878 human curated SNPs in Opus are not the only SNPs that Opus looks at. Opus has access to much bigger libraries of SNPs, including the Genome-Wide Association Study SNPs, the ClinVar SNPs, pharmacogenomic SNPs. These SNPs are the ones that are human curated with particular reference to their utilization in a nutrigenomically driven practice. So these are basically going to be the SNPs that are going to have prescriptive elements that would be reflective of a functional or naturopathic type practice. And essentially at that point you've uploaded the client, and then you can work with the client at that point. So at some point in the distant past, I uploaded this particular person. Opus told me it was 23andMe V4, and this is the coverage that was theirs. So once the person is live, you'll see all of a sudden a whole bunch of options appear up in this upper menu bar. We're not going to talk about all of them in this particular video, but we're going to talk about a few basic functions in Opus that are key to getting the most out of the program. And in order to do that, I'm going to go to the first option here with regard to analytics, which is called mappings. And these are pathway maps that are animated with client genomic outcome data. And let's go find a simple example of this. Let's go to the maps that are associated with gene expression. And you can see here we have two maps that have been tagged as being significant with regard to gene expression. And certain parts of these maps are colored, which we'll get to in a second when we click on a specific map to bring that up in the map examination window. So let's click on vitamin D mediated expression. Here we have a map of the pathways that are involved in the actions of the vitamin D receptor and vitamin D supplementation. This map is a scalable graphic, so for instance, if it doesn't look big enough to you, you can hit this button down here and increase the scale, or you could reset it. Let's make it a little bigger, but also too, if you go into the screen and hold down the mouse key, you can drag it so that you can center it on areas that are going to be significant to you. Now, the important thing here is to understand essentially what's being displayed. And to understand that, I think we have to take a step back to understand how the whole hierarchy of this information is organized. You probably well know that there are single nucleotide polymorphisms, which represent a specific place on the genome where a particular set of alleles can be found. And when there are polymorphisms at that location, it means that there can be situations where the actual letter that represents the specific nucleotide could be variable in certain populations. So if I go to location 10,047 on chromosome 19, and 90% of the world has a T, and 10% of the world has a C, it means that that particular location has a polymorphism, and a percentage of the population has the major allele, and a percentage of the population has the minor allele. Also to keep in mind that nucleotides as reported by genotype are a combination of two alleles, one from each parent. So with the exception of sex-linked chromosomes, most every location, SNP location, is reported as two alleles. So we might say that person so-and-so had at location 10,417 was genotype CT. Meaning that they had one allele for the cytosine and one allele for the thymine nucleotide. Well, that basically would imply that there was some significance, there was variation recognized. However, the person was heterozygous, which means that they had one allele for perhaps the allele of interest and one allele for the normal allele or the non-significant allele. So what do we look at when we see that? Well, let's say we set it at location 10,452. There was a risk allele for myopia that was associated with that polymorphism. The risk allele is T. If a person basically types genotypically as CT, they're heterozygous for the risk allele. If they type as TT, they're homozygous for the risk allele. Generally, in this type of work, homozygous for the risk allele is much more kind of attention getting than heterozygous for the risk allele, because a lot of times the heterozygous state still conveys significant functionality to the gene as it's being able to express a workable functional protein, typically an enzyme. So if we're looking at these kind of things, we would see homozygous for the risk allele would be much more attention. So when we look here, when we look at this map, we're actually looking at something that's depicting the level of reporting for a specific gene or molecule as a result of the sum total of all the polymorphisms that have been linked to the gene and their significance. To understand that, we would click on a particular gene here and it would bring up an information pop-up. But before I do that, I just want to draw your attention to the fact that you'll notice some of the boxes are colored yellow, some boxes are colored gray, and some boxes are colored a kind of a dark orange, and are colored in various degrees of filling the box. Now, that's a mathematical representation of the status of the SNPs that are being linked to the gene and their outcomes. To understand that, we want to basically understand that a SNP has a certain level of magnitude. There are certain SNPs that have highly significant magnitude, for instance, like BRCAs and all sorts of other SNPs, where a single mutation is significantly associated with risk of pathology or ApoEs or whatever. That's in Opus considered to be a power factor. So a SNP that has a power factor of 8 is a significant SNP because the range of power factors goes from 1 to 10. If you've ever been to SNPedia, they do a similar thing. It's called magnitude, but in Opus it's called power factor. Power factors are determined by algorithms that have to do with the weight of the studies associating the mutation with consequences biologically, and the prevalence of studies to support those associations plus the effect. So in other words, when the editor is actually curating the SNP, they're going through a process of giving that SNP a certain level of significance. So when we look, in this case, let's take a look at the VDR receptor here. You can see here if I hover over it, it'll tell me that it's going to report 30 SNPs. And a significant number in this client of power factors have been found to be associated with a homozygous for the risk allele, which is the orange color. In this case, if we were to go over to tumor necrosis factor, we would see that a significant number of the total number of magnitude or power factors of SNPs that we link to tumor necrosis factor, a significant amount are comprised of heterozygous outcomes, and that's represented by the yellow color. In the case of, let's say, a SNP that's being reported as a gray color, it means that there's no recognized mutations of significance that have been associated with the SNPs that we monitor with regard to that gene. So in the case of, let's say, a person with the ApoE here, we would find that there was a quick snapshot of this tells you that there's areas there that could be not necessarily very significant. On the other hand, looking at, for instance, interleukin 12B, we would see that of the total number of power factors linked to all the SNPs combined that have been linked to the SNPs that we associate with that gene, there's nothing but homozygous risk allele outcomes. And so, again, it's just a very quick way to get a snapshot of a gene that's basically giving you some insight from a kind of a visual perspective. I like to think of this as like the power bar on your phone, that tells you how much battery life you have left. When you start talking about the sort of the badness, extends this way. Uh, again, it really brings up one other thing. We're going to be referring again and again to these maps in the course of our other films. But the most important thing I wanted to tell you was how everything in Opus is driven by information pop-ups. So let's click on the VDR and you can see how it works. Here's an Opus pop-up. This is basically an education in nutrigenomics and systems in molecular biology. These particular entries are curated by the editors, and these represent some of the most advanced uh, insights in terms of informational analytics, data information, with regard to what we really want to understand about this particular location. So you'll see here that I'm scrolling down the pop-up and there's just a huge, well, VDR is a very significant player. And when we get down to here, you're going to see many things actually show up as being represented in the pop-up. These pop-ups, as I said before, you'll spend a lot of time looking at these because they serve to they're like your assistant when it comes to being able to find what is going to be significant. And of course, not many of us walk around with a working knowledge of 40,000 genes in our head. So these things really become something along the lines of almost like a flashcard type of situation. The more you open up the pop-up on VDR, you might the first time learn a few things. The second time you'll remember more. So actually, as you work in Opus, it has one of these kind of vicarious side effects of really essentially being a kind of a training in genomics and precision medicine. Just by the usage of the program just makes you more knowledgeable and smarter. So you see here that the pop-up is showing that the VDR is represented in a particular algorithm. Now those are things we'll talk about in another video. But algorithms are just logical consequences of multiple SNPs. So in other words, very few SNPs are unique in and of themselves. That most of the time in genetics things function as what are called haplotypes, where a SNP here and a SNP there have a kind of a Boolean relationship where if this SNP is T and this SNP is not C, then stand on your head and rub your stomach. So the reality of it is that we do a lot of work where we analyze multiple combinations of SNPs to be able to draw outcomes, and those have oftentimes real-world significance. And you can see here we have an algorithm that involves the vitamin D receptor in the preferred forms of B12 supplementation, which we'll get to when we go through the whole algorithm video, which will be probably 2 or 3 videos from now. But clicking on these pop-ups are just a phenomenal way of being able to acquaint yourself with the way Opus works. You can see here, this is the inventory of all the SNPs that we're actually watching in Opus that are linked to the VDR. Here's the SNP, here's the power factor, here's some information with regard to various tags or labels that are associated with it. This is the interest allele, which is the allele that is either the risk or benefit. It's the allele that has something that somebody discovered about it makes it important. So let's take a look at here. We can see that the TAC1 SNP has a power factor of, it looks to me like about 8. The interest allele is G, and this client is homozygous for the risk allele. Here's another high value SNP here, where the client has the homozygous outcome for the risk allele T. And you can deep dive into these pop-ups. For instance, if at this point I need to learn a little bit more about the SNP itself, I can go ahead, and now I'm not reading about the vitamin D receptor anymore. I'm reading about the SNP that is part of the family of SNPs that are linked to the vitamin D receptor. And you can see here there's HapMap data in terms of frequencies that are expected in the population. Some data that's giving you some of the information that the curators and the algorithms have determined. It's got a very high level of curation, meaning that the curators are constantly into this SNP because of its clinical significance. It's got a very strong evidence basis, and it's got a very strong power factor. So again, all the things that here are part of the reporting are things that, they're sort of important, I guess. Generally, you're getting some information on the DB SNP orientation. SNPs, as you might know, come in what are called strand orientations, depending upon which way the strand is read. Either you can have the minus strand or the plus strand. They're kind of they're complementarily linked. So if it's a G on the minus strand, it's going to be a C on the plus strand. So in other words, it's kind of the inverse. However, almost all the report, this is where things get a little kinky. All the reporting services always report its genotype outcomes on the positive strand. But a lot of the clinical services, because researchers use machines that can report on either strand, so the research services like ClinVar or GWAS or DB-SNP, sometimes they report on the positive strand, sometimes they report on the negative strand. But Opus is telling you in this case that the DB-SNP orientation was minus, and how Opus actually just converted it so that Opus always converts everything to the positive strand because that's the way it gets the data from the reporting services. Again, now we're talking about specifics on the SNP. We're talking about specifics with regard to those algorithms. There's nutrients that basically are associated with the proper function of the SNP. In this case, magnesium has some information that it would be a ability to activate or improve the function of the protein that typically the enzyme that its activity is inhibited by the risk allele. And then again, we have a variety. Opus has a tremendous inventory of natural products in terms of the natural product databases, as you'll see. And what we're seeing here is not necessarily information with regard to the VDR receptor. I mean, to the particular SNP. Now it's giving you some information that goes back to the gene. These are related to the gene itself. And in this case, we're seeing here that, well, not surprisingly, vitamin C is an agonist of the VDR receptor. Next to it, you see here that there's some PubMed references that are also part of this. And this is important. There's a lot of hooey in a lot of the functional medicine genomic and nutrigenomic associations. People say things. If you want and you trust that person, you can take it at face value. Perhaps people share clinical experience about things that has some value. However, Opus only works on one basic premise. You better have a reference in PubMed to support what it was that you're actually making a claim about. So everything in Opus always drills down to the original study. And even in Opus, you can actually go and find that particular study that basically is used to be able to make that association. So this is the great thing about Opus. Everything original, and no matter how high or how low you go through these informational pop-ups, it always comes back to something that you could double check to make sure that it was representational of what it was that we were implying in what we shared as information. So you can always close a pop-up by just clicking. You can click on the X, but you can click outside the box and it goes away. So again, looking at let's find VDR again because I want to go to the bottom of the screen here because I mean this pop-up is obviously this is a very important SNP, so you're going to have a very extensive pop-up. But here's an interesting thing. This is a radar chart of what you would call the agent expression logo. So we're looking here at the informational display of those natural products with regard to their efficacy in terms of the studies. Because this radar part's a little eccentric because vitamin D is like an 800-pound gorilla here. But it's normally, if you've got something that doesn't have such a big difference between like the 800-pound gorilla and everything else, this would be very much more illustrative in an informational standpoint. However, you've got this information just down here as well. And so, for instance, if we need to look and see, okay, well, we've got some evidence here because it's ranked as a 2 that magnesium is an agonist to vitamin D receptor. So let's go see what magnesium is. So you can see how you can navigate in and out of these pop-ups and just really get a first-rate education in the natural products, pharmacognosy, herbal medicine, nutrigenomics, lifestyle. It just comes in everything. You can zoom in, you can zoom out. But I was saying before, you can also, in addition to clicking out of the pop-up, you'll often see a button here that says go back, and you'll take you back to the prior pop-up as well. So what do we notice is that there's a point where we see a new Opus doc takes the training and starts using Opus, and they buy a license for themselves and for their brother or their mother, and then we don't see any activity. They don't really buy a license for, like, I don't know, a couple of weeks, and it's like, oh, maybe they didn't like the program. But every time we inquire about it, it's that they're spending that 2 weeks just using Opus as a sort of a training tool for themselves. They put those first two or three people in, and then they're just working in Opus with regard to being able to get better at the program. But also too, I mean, this is a terrific way to nibble at an extraordinarily complex, wide-ranging, enormous field. And you just can't approximate that all in one day. You have to nibble at it on a day-to-day basis. And eventually, repetition becomes reality. And over time, you become so much more better at being able to draw conclusions from things. So, what we've actually discovered in this particular video is, number one, how to get into Opus, how to log in, how to load a patient into Opus, load a client into Opus, and to begin to work in the most basic element in Opus, which is looking at pathway maps, and then discover the amazing capacities that are inside the pop-up windows with regard to be able to improve our understanding and knowledge. We also have explored at least one map here, vitamin D, but let's go take a look at another one. Let's go take how about detoxification, shall we? Here, let's go look at glutathione. Here's a map of phase 2, glutathione conjugation and sulfation, and we can see here that perhaps maybe we need to do some thinking about doing some work with glutathione reductase, and perhaps maybe doing some augmentation of some of the glutathione peroxidases. As you know, the phase 2 glutathione conjugation and sulfation is also dependent on how effectively phase 1 was able to get the job done. So again, we would be looking at our informational pop-ups that would be taking us, okay, why is this gene so compromised? Well, because the four major SNPs we follow, the patient has got homozygous for the recessive. And here's a very well-functioning logo where you can see there's not necessarily an 800-pound gorilla. And you can see here that sometimes you wind up in situations with data that just tells you just how gnarly things can be. When we see a green color here, it's telling us that this substance is a agonist of this particular gene. When we see the orange color, it means that this substance is an antagonist. It's been studied to show that it has some effects of lowering function. And occasionally you get into that bizarro world where sometimes you've got data that actually conflicts. What do we know about that? Well, the nature of how you arrive at truth is over time the weight of the evidence. And sometimes you wind up with mixed results. And again, if you really feel like you need to find out why they came up with two different types of outcomes, well, you can click on number 2. And this should be able to give you some insights as to why things could be so mixed up. So really, I think the important thing in this lesson was get into Opus, get a few clients in Opus, make sure you get the right data, get enough data in there to do some stuff. Take a quick look at the maps with insights that you can garner from the color coding. Fire up a few pop-ups to start to look at things that you may be familiar with that you need refreshing for, or on the other hand, click on things you never heard of and see if that makes a big difference in terms of your understanding. Looking at the maps, try to understand how the pathways are basically represented. Print them out. There's all sorts of opportunities for you to be able to look at this from a perspective of how to get around. The thing that you'll learn about Opus is it's an amazing program in that it doesn't do anything a certain way. It does the same thing multiple ways. And the reason for that is sometimes if you ask a question two different ways, you get slightly different answers. And sometimes being able to triangulate between the differences in those answers gets you closer to reality. You get a better approximation of what reality was. One of my inspirations said you get a piecemeal approximation of reality through these generative mechanisms. And you're going to see that there's a plethora of ways that you can parse data in Opus and find out all sorts of insights that really, if you just only had a simple way of looking at things in a table, oh, the snip is bad, I'll make that box red, or so and so. You get a cartoon version, which maybe is actionable, but really, you haven't approximated reality. So what Opus tries to do is give you multiple ways of being able to ask a question that you could get different answers that may be illustrative of the fact that reality is lying somewhere in between. And you'll see as we go through all the different apps, it's a very powerful way of interrogating data. Well, this is the end of the first video, and I'm going to sign off now, and hopefully you'll have some materials in this lesson that you can work with. And again, welcome to Opus. It's really an exciting program. You'll see as we get further along. It can do an amazing amount of things. It's beautifully designed for telemedicine education, and I'm so happy that you chose to trust us with this important information on your clients. So again, welcome, and let's get started learning about Opus and getting it into our practice and lives.